Swayam Prabha. Digital India. Educated India.
Welcome to my course on genome editing and engineering. We are going to discuss about basics of zinc finger nucleases today. So, we will be first learning about some of the basic things which are important for this lecture. We will learn about the central dogma, the protein structures and how proteins bind to DNA. Then we will be discussing about one nuclease called FOC1 nuclease and certain special motifs in proteins called zinc finger domains. So, this figure may be familiar to many of you, which depicts the central dogma of molecular biology or central dogma of life, where DNA is the information molecule which is copied by RNA. You can see in this arm by the method of transcription, and this RNA is translated into proteins. Also, the DNA replicates on its own or make copies of itself by the process of DNA replication, and there are also methods by which RNA may be copied into RNA, but we are not going to discuss much about those molecular mechanisms today. We are going to use some of the concepts of protein structure important for this lecture. You know about the primary structure of proteins, which are basically linear polymers of amino acids, and the secondary structure elements are there known as beta sheets and alpha helix, and then the tertiary structure which is basically the overall 3D folding of proteins or the arrangement of the secondary structures in space, and that makes a protein functional or active. And then you have the quaternary structure which is basically the arrangement of the tertiary structures with respect to one another in 3-dimensional space. This is one of the landmark papers in science by Linus Pauling, Robert Corey and Branson, where they described some of the important elements of protein structures. They came to a conclusion that linear polypeptide chains in the primary structure fall into regular repeating structure units, and they name these repeating structure units as alpha helix, which are basically a spiral arrangement of the amino acids in 3D space, and the second structure as beta pleated sheets. The name alpha and beta is due to the order of their discovery, and they constitute the elements of protein secondary structure along with other elements like the loops, turns and the hairpins. Let us focus a little bit about the alpha helix, and this is very important from the point of view of studying zinc finger domains. Alpha helix is the most common type of secondary structural element in proteins. For every turn in these helix, there are around 3.6 amino acid residues or one residue per 100-degree rotation. These residues translates 1.5 angstrom along the helix axis and giving a vertical distance of around 5.4 angstrom between structurally equivalent atoms in a turn, or the pitch is something around 0.5 nanometers. As you can see, this is a rod shaped structure, and this rod-shaped alpha helix structure is stabilized by hydrogen bonds between NH and CO groups of the main chain. The NH group of an amino acid forms a hydrogen bond with the CO group of the amino acid 4 residues earlier. So, there is a repeating pattern of I plus 4 to I hydrogen bonding, and it is the prominent characteristics of an alpha helix. Next are the beta sheets or beta strands. A beta strand is an element of secondary structure in which the protein chain is nearly linear. Here there is no any rod-shaped structure like the alpha helix. So, 2 or more adjacent beta strands will bind to each other through hydrogen bonding to form a beta sheet. These are also called as beta pleated sheets. The participating beta strands are not continuous in the primary sequence and may not necessarily have to be close to one another in the sequence. The strands forming a beta sheet can be separated in primary structure by long intervening sequences of amino acids that lie outside of the sheet structure. Now, these beta sheets may be arranged in different orientations. One of is the parallel beta sheet orientations. Here all bonded strands have the same N to C direction. As a result, they have to be separated by long sequence stretches. The hydrogen bonds are equally distanced. The figure to the left, you can see the 4-stranded parallel beta sheet from a protein. So, let us discuss some of the various orientations of the beta pleated sheets. The first one being the parallel beta sheets. All bonded strands have the same N to C direction. As a result, they have to be separated by long sequence stretches. The hydrogen bonds are also equally distanced. In the figure in the left, you can see a 4-stranded beta parallel sheet from a protein. The 4 parallel strands are shown in cartoon format. The hydrogen bonds are identified by the dotted lines containing the donor nitrogen and the acceptor oxygens. Then you have the anti-parallel beta sheets, and you can see these, the direction of the beta strands are opposite to one another, but they are parallel. And here the beta strands run in alternating directions and therefore can be quite close on the primary sequence. The distance between successive hydrogen bond alternates between shorter and longer in this case. And on the right, you can see the figure of a 3-stranded antiparallel beta sheet. The 3 antiparallel strands are shown in cartoon format and in the stick form. Hydrogen bonds are identified by the dotted lines connecting the donor nitrogen and acceptor oxygens. Then there is a third type of arrangement which we call as the mixed beta sheet. This is basically a mixture of parallel and antiparallel hydrogen bonding. About 20% of all beta sheets are found to be mixed beta sheets. Now we know about the central dogma of molecular biology. You also know about the various secondary structures of protein as well as the tertiary structure and the quaternary structure in brief. Why do we need to know this? We have to know that DNA is the informational molecule. Except storing and providing information, it cannot act on its own. The work is being carried mostly by proteins and RNA, and RNA copies these information from DNA and translates them into the executors or the proteins. And in certain cases, the RNA molecule is also a functional molecule. RNA may have a structural role, RNA may have some kind of catalytic role, but the role which is being played by protein is highly diverse when we compare it with the roles being carried out by RNA. So, for doing all these work, protein needs to talk to all other molecules inside the cell. Protein has to talk to DNA, it has to talk to RNA. So, protein has to bind to DNA for whether replication to be carried out or transcription to be carried out or any other activity related to DNA. Similarly, proteins also bind to RNA and then carries out translation, and besides that, proteins also binds to proteins themselves, or there are protein-protein interactions. So, proteins are known as the players which drives cellular events by binding to DNA, RNA and proteins themselves. So, we now know that the proteins bind to DNA and RNA. So, there are proteins which we can call as DNA binding proteins, and this study has reported that we have roughly around 588 DNA binding proteins, and we have a large number of RNA binding proteins, which is more than double of the DNA binding proteins, being 1210 roughly. And there are some proteins, a small set numbering something around 64, which binds to both DNA and RNA at the same time. So, this study is from the QuickGo database, and this is the reference which you can consult for knowing more. So, already a little has been told to you about the importance of proteins in driving the cellular events. Now, let us focus on the DNA binding proteins, which play a central role in all aspects of genetic activity within an organism, such as in transcription, packaging of DNA that we have discussed at length in the introductory classes, then rearrangement of DNA that also has been discussed while studying homologous recombination, any and etc., and replication and repair. So, a study was carried out to analyze around 240 protein DNA complexes or of DNA binding proteins, and all these yielded around 8 different structural functional groups, which were classified further into 54 structural families. So, basically these 8 large groups of protein structural functional group are the helix turn helix, the zinc coordinating proteins, the zipper types, then other alpha helix and beta sheets, then beta hairpin ribbon, then a miscellaneous group having diverse functions with some commonalities, and there is age group, the enzymes. Some of the enzymes also fall into the other 6 classes. In this particular lecture, we will focus mostly on 2 protein structural or functional groups. They are the alpha helix turn helix group and the zinc coordinating group. Let us first start with a discussion on the group 1, which is the helix turn helix. So, as you can see, there are many helices over here. These are basically the alpha helices which we discussed in the beginning of this lecture. So, this HTH motif or the helix turn helix motif is a common recognition element in proteins used in transcription regulation and enzymes of prokaryotes and eukaryotes. This is an important point we need to carry forward along with us for the remaining part of the lecture. Here you can see the helix turn helix DNA binding domains in lambda phase CII transcriptional activator. Let us now move on to the next part of this lecture, the fork 1 nucleases. Let us now discuss about the fork 1 nucleases, which are important players in the ZFN technology platform. Flavobacterium echinocoides was first reported by Jobel and Upham in 1944, which is a gram negative non-motile and motile Rothschild bacteria. It occurs in soil and fresh water in a variety of environments. We just discussed about the helix turn helix group, structural functional group. This particular organism, Flavobacterium echinocoides, produces an endonuclease called fork 1, which is a member of the unusual type 2 class of bipartite restriction endonucleases that recognizes a specific DNA sequence and cleave non-specifically a short distance away from that sequence. This is one of the very important discoveries in these particular field where Hiroyuki and his associate discovered a new restriction endonuclease from Flavobacterium Echinocoitis fauquon and another one from Micrococcus luteus. So, in this paper you can see from the abstract the importance of these two clones which produces these two type of endonucleases, restriction endonucleases, and here the fauquon belongs to a class of restriction endonucleases that recognizes specific but asymmetric nucleotide sequences and introduces staggered cleavages at the pointed positions away from the recognition sequences. So, these are not cutting and recognizing at the same place, and they produce blunt ends or staggered cleavages. In the discussion of this paper, the authors write about one observation where cloning vector PBR322 does not contain the amylu1 site. Therefore, this enzyme could become a useful tool for gene manipulation. Okay? And they could not anticipate the potential of fauquon much, and they concluded that fauquon should be convenient for construction of accurate cleavage maps of DNA because fauquon cut non-specifically. So, with this, however, the scientific world came to know about the existence of this particular enzyme fauquon, and soon it was forgotten. The discovery was sometime around 1981. So, this is the binding sequence of the fauquon which binds cognate sequence 5 prime GG ATG here and cleaves DNA phosphodiester groups 9 base pairs away here. So, it will bind here and it will cleave here. On the 5 prime strand and 13 base pairs away on the 3 prime complementary strand, and it will give a staggered cut as a result of this reaction. The molecular mass of this enzyme is around 65.4 kilodalton, and it contains around 587 amino acids, and as already told to you, this is a type 2 restriction in endonuclease, and it consists of 2 functionally distinct domains. Number 1 domain is the N-terminal DNA recognition domain and a C-terminal DNA cleavage domain. So, the domain which is in the N-terminal will bind to this cognate sequence, and domain at the C-terminal will cleave at the 9 base pairs to 13 base pairs away. Now, cleavage occurs only upon dimerization. If only one fauquon enzyme binds to the DNA, it will not cleave. It needs a partner on the complementary strand as well. So, when the recognition domain is bound to its cognate site and in the presence of magnesium ions. So, these are some of the requirements for this enzyme to function. So, as you can see that it has its 2 modules, one is the N-terminal domain and another is the C-terminal domain. The modular nature of fauquon has led to the development of artificial enzymes with new specificities, which we are going to study at length in the next lecture, and a little bit of that in this lecture too. Now, this is one of the important work carried out by Chandrasekharan and his colleagues, and they published their results in 1982, where they identified the functional domains of fauquon restriction endonucleases. So, their study proved the presence of 2 separate protein domains within this enzyme, which we have already discussed. One is for the site specific recognition and another one for the endonuclease activity. So, this recognition domain as you can see may have several subdomains D1, D2 and D3, and there is a cleavage domain in the N terminus, and this is connected through a linker molecule as shown in this particular picture cartoon. The fork 1 exists as a monomer and first binds to DNA as a monomer. This complex is catalytically inactive. So, first time this particular monomer will bind to a DNA strand, okay, as a monomer, and then another fork 1 will bind on the other strand later. Structure of the fork 1 dimer. Dimerization is mediated by the parallel helices alpha 4 and alpha 5 of the cleavage domain. There are no significant interactions between the D1 subdomains despite the appearance of such in this orientation of these structures. So, they remain further away from one another and they do not have any role in this dimerization, but the domains the alpha helices 4 and 5 in this cleavage domain play a major role. A fork 1 monomer first binds DNA at its recognition site, then a second fork 1 molecule bound to another recognition site would dimerize with the first molecule through their cleavage domains. The catalytic domain of each fork 1 molecule swings away from the recognition domain to position its catalytic sites opposite to the targeted phosphodiester bond and catalyzes cleavage at the first site. After cleavage, the two fork 1 molecules dissociate from the DNA. As already told to you, the dimerization is primarily mediated through parallel helices alpha 4 and alpha 5 and two loops P1 and P2 of the cleavage domain. The two alpha 4 helices are roughly perpendicular to each other, and together the two helices make up the core of the dimer interface. The residue ASP 483 of alpha 4 makes bidentate hydrogen bonds with ARG 487 of alpha 4 of the other monomer and vice versa. The core is sandwiched between the helix alpha 5 and loops P1 and P2 from each monomer, where loop P1 comes between the beta strands beta 1 and beta 2 and loop P2 is at the C terminus of alpha 4. So, let us now move on to another important concept required for the ZFN technology, the zinc finger domains. Let us now discuss about the zinc finger domains, which are very important for understanding the ZFN technology. The biological role of zinc in proteins was established in 1940 and it was shown to participate in carbonic anhydrase catalytic activity. Soon it was found that the zinc ions play a unique role not only for CA but also other enzymes catalytic activity. In 1985, the structural role of zinc was proposed for the Genopus levis transcription factor 3A or TF3A. The Genopus levis TF3A structure consists of repeating 30 amino acid residues folded around the zinc ion to form a small highly independent domain called a finger. Around 20 to 30 residues form a loop during zinc binding to conserved cysteine and histidine residues, while the other 5 residues form linkers between conjugative fingers. Moreover, for each repeat, the special conserved pattern with 3 conserved hydrophobic amino acids tyrosine 6, phenylalanine 17 and leucine 23 was also reported. Let us focus on some of the features of zinc coordinating proteins. The beta, beta, alpha zinc finger proteins together form the largest individual family in the group. There are over 1,000 plus distinct sequence motifs which acts as transcription factors The structure of a typical zinc finger includes a short two-stranded antiparallel beta sheet and an alpha helix. So, you can see here the beta sheets are antiparallel and it has a alpha helix over here. Two pairs of conserved histidine and cysteine residues in the helix and beta strand coordinate a single zinc ion. Zinc fingers are encoded by approximately 3% of the human proteome and are the most common DNA binding motif in eukaryotes. Transcription factors often use zinc fingers to mediate protein DNA interactions. Krishna and associates classified all zinc fingers into 8-fold groups, and you can see here the 8 different fold groups over here and having given different names. The C2H2 like finger, gag knuckle, treble clef, zinc ribbon and so on. Amongst these you can have many family members for each fold group. For example, 2 of the family members that we have mentioned here are the C2H2 finger family, IAP domain family, and in the gag knuckle you can see a couple of families being listed over here, and accordingly also in the treble clef you have the ring finger like protein kinase cysteine type domains and so on. Amongst all these 8 folds, the C2H2 like finger, the treble clef finger and the zinc ribbon finger constitute the majority of the zinc fingers available in the biological world. These zinc fingers were first identified in a study of transcription in the African clawed frog Genopus levis, as already told to you earlier, in the laboratory of Aaron Klug. And this is the landmark paper Aaron Klug and his co-workers Miller, McLachlan published in the EMBO journal in 1985. You can refer to this paper for full details of this particular discovery. Briefly, in this paper, a study of the transcription of a targeted RNA sequence revealed that the binding strength of a small transcription factor TF3A was due to the presence of zinc coordinating finger like structures. To know about these zinc coordinating finger like structures, amino acid sequencing was done, and the amino acid sequencing of these particular TF3A factor revealed 9 tandem sequences of 30 amino acids, including 2 invariant pairs of cysteine and histidine residue. Interestingly, in this work they did not use much of computational power, and these repeating motifs were detected by the eye, scanning by the eye and not by computer analysis. So, it is very important to know some of the discoveries being done in very simple ways, and these are the words of Aaron Klug. So, I should however emphasize that the repeating structure was discovered through biochemistry and not as some reviews have stated by computer sequence analysis. When the sequence was published, I looked for and found by eye a repeating pattern which was then confirmed and aligned as a motif of 30 amino acids by Andrew McLachlan's computer analysis. C2H2 finger family. So, you have 2 cysteine and 2 histidine as signature amino acids in these particular families, and since its discovery in the Genopus levis transcription factor 3A, the C2H2 zinc finger motif, also referred to as the classic zinc finger, has been found to be present in many transcription factors and in other DNA binding proteins which recognize specific sequences of DNA. A typical C2H2 zinc finger contains a repeated 20 to 30 amino acid sequences, including 2 conserved cysteine and 2 conserved histidine residues, but other combinations of cysteine and histidine is possible. For example, in gene chelating as gene chelating residues. The regulation of the transcription seems to be the most important task performed by the C2H2 zinc fingers. However, recent studies have suggested their roles in mediating protein-protein interactions as well. So, we now know that in Genopus levis they had found around 30 amino acid residues, but further studies reveal that their C2H2 domain has a range of around 28 to 30 amino acids, and these C2H2 domains include certain secondary structure elements, a beta hairpin which comprise of antiparallel beta sheets consisting of 2 beta strands. So, 2 beta strands in antiparallel orientation give rise to a beta hairpin, and which is a important component of this classical C2H2 domain. Then it has, in addition to this, an alpha helix, and together they form a left handed beta beta alpha structure, 2 beta coming from here and 1 alpha coming from here, and the beta hairpin and the alpha helix. The zinc finger structure is stabilized by coordination of a zinc atom with 2 conserved cysteine residues at one end of the beta sheet and with 2 conserved histidine residues at the alpha helix C terminus. Not only the cysteine and histidine pairs are conserved, there is a highly conserved hydrophobic core forming the alpha helix. The other amino acid residues in C2H2 domains are largely variable. In all cases, zinc ion plays a structural role and does not interact directly with the DNA. The alpha helix serves as the recognition element inserting into the DNA major groove, whereas side chains form specific contacts with 3 consecutive base pairs. The overall interaction is further stabilized by non-specific contacts between the beta sheet and the sugar phosphate backbone of DNA. The zinc finger domains are among the most structurally diverse protein domains. They interact with nucleic acids, other proteins and lipids to facilitate a multitude of biological processes. Zinc fingers are categorized into various classes and have various architectures, metal binding modes, functions and reactivity. So, here you can see the structure of C2H2 zinc finger, and the zinc ion, which is shown in green, is coordinated by 2 histidine and 2 cysteine amino acid residues about which we have discussed in the previous slide. The versatility, selectivity and stability of these short amino acid sequences is achieved mainly by 3 things: residue participating in zinc coordination, mostly by cysteine and histidine, then hydrophobic core and ZF structure formation, and variable residues responsible for inter and intramolecular interactions. So, this is the thumb rule which you may remember to understand the zinc coordination. If you put your wrist in this orientation with the thumbs up, then in this side you have the 2 cysteine residues, and on these 2 fingers you have the zinc residues, and in your palm below the thumb you have the zinc molecule, and this is how they bind and stabilize the ZF structure. Sequence alignments of the C2H2 zinc fingers and other studies have revealed that the residues that are responsible for the hydrophobic core formation in most C2H2 zinc fingers are L7, F11 and L2, and this is a schematic of C2H2 zinc finger domain primary structure with the bones coordinating the zinc ions and a drawing of hand and finger background. The cysteine and histine are the coordinating residues respectively. A canonical zinc finger module consists of two antiparallel beta strands and an alpha helix with a zinc ion coordinated by the conserved cysteine and histidine residue pairs. Various amino acid residues at positions relative to the amino terminal end of the alpha helix form important base specific contacts in the major groove of the double stranded target DNA. The zinc finger domain is one of the most frequently utilized DNA binding motif found in eukaryotic transcription factors as well. The binding of a zinc finger domain to its target site juxtaposes 3 base pairs on DNA to a few amino acids in the alpha helix structure. The identity of the amino acids at the contact site defines the DNA sequence recognition specificity of the zinc fingers. So, you have here 3 fingers, finger 1, 2, 3, and you have some cognate sequences which are in the DNA. So, basically a ZFP consisting of 3 link ZF modules are being shown in this cartoon, and it binds its target DNA site with amino acids of the recognition alpha helices in the N to C terminal direction, contacting consecutive nucleotides in DNA running in the 3' to 5' directions. This can sometimes lead to confusion because the DNA target site is typically referred in the other direction, which is the 5' to 3' direction. Let us now discuss how the proteins recognize DNA sequences and bind to them. The DNA binding proteins recognize DNA sequence with at least 2 different binding modes, specific and non-specific modes. DNA binding proteins would initiate gene transcriptional regulation by searching for their target DNA sites among an overwhelming number of non-specific DNA sequences in the nucleus. Theoretically, the protein first binds non-specifically to any given sequence of DNA and then rapidly searches the sequence for the presence of specific binding sites. In doing so, it sometimes would move along the DNA strand to find for its cognate sequences. So, briefly, the protein DNA recognition involves at least 2 steps. The first step is the non-specific binding. The protein would bind to any given sequence of DNA without bordering for the target or the cognate sequence. So, firstly, the DNA binds to any available DNA sequence to form a non-specific protein DNA complex, and in these non-specific reaction, the electrostatic interactions are the important dominating factors. Later on, it gradually would shift towards specific binding. So, following non-specific binding, a protein will slide along the DNA backbone looking for its cognate sequence. In this process, the protein interacting residues can switch roles from a purely electrostatic interaction with the DNA backbone in the non-specific complex to a highly specific binding mode with the base pairs of the cognate sequence. So, here is a model of the site specific DNA recognition by C2H2 zinc finger domains. So, in figure A, you can see a crystal structure of 3 zinc fingers, 1, 2 and 3, and this is the DNA molecule in the center. The amino acids involved in the site specific DNA recognition are color coded. -1 is green and +2 is blue plus 3 is red, and plus 6 is purple. On the corresponding panel, figure B is a model of the site specific DNA recognition by alpha helical amino acids. A model of the site specific DNA recognition by C2H2 finger domains is being shown in this figure. Figure A, you can see the crystal structure of 3 zinc fingers of the GIF268 protein bound to DNA. So, the amino acids involved in the site specific DNA recognition are color coded, -1 green, +2 blue, +3 red, and +6 purple, and these are the 3 zinc fingers which are binding to the DNA molecule in the center. In panel B, you can see a model of site specific DNA recognition by alpha helical amino acids. So, here you can see +6 corresponding to 1, +2 corresponding to 2, +2 corresponding to 3, and 4, and -1 corresponding to 3. So, we will discuss about this relationship in a later slide. So, this is one of the important paper in finding out how zinc fingers recognize DNA molecule, and this work by Pavletic and Pabo were the first to determine the crystal structure of a complex of DNA with 3 tandem C2H2 domains of the mammalian GIF268 proteins. The 3 zinc fingers were found to form a semicircle located in the major DNA groove. So, in this complex, as you can see in their abstract, the zinc fingers bind in the major groove of beta DNA and wrap partway around the double helix. Each finger has a similar relation to the DNA and makes its primary contacts in a 3 base pair subsite. Residues from the amino terminal portion of an alpha helix contact the bases, and most of the contacts are made with the guanine rich strands of the DNA. And their work provided a framework for understanding how zinc fingers recognize DNA and suggests that this motif may be useful basis for the design of novel DNA binding proteins. So, each of the 3 C2H2 domains bind to 3 or 4 DNA nucleotides via amino acids at the same alpha helical position. So, these amino acids positions are in the alpha helix. So, they are in the positions -1, +2, +3, and +6, and these are the positions of the DNA nucleotides in the forward strand and in the reverse strand. As you can see, 5 prime, and this is the type 3 prime. So, arginine at position -1 as well as the amino acid residues at position 2, 3 and 6 respectively take part in this interaction. The biochemical and structural studies of these C2H2 domains confirm the key role of the amino acid at these positions for the specific binding to DNA, and there is a canonical model which suggests that the amino acids at the positions 6, 3 and 1 are responsible for recognition of the first, second and third nucleotides at the 5 prime end or the forward strand of the DNA respectively, and these 6, 3 and -1 are responsible for binding to 1, 2 and 3 nucleotides in the forward strand or the 5 prime strand, and the amino acid at the second position binds to the 4th nucleotides on the complementary strand. So, this is very important to remember how the zinc fingers C2H2 domains bind to a double stranded DNA molecule. It is not just the 3 bases on the 4th strand, a 4th base also sometimes play an important role, and this 4th base is interacted by the 2nd amino acid residue. Now, we studied about the binding modes, how it starts with a non-specific binding in the beginning, and then it the complex slides along the DNA molecule, and then upon finding its cognate partner, it transforms into a more specific binding interaction. So, this is a proposed sequential binding scheme of 3 cis2 his2 zinc finger domain or TJD bound to the cognate DNA, and this work was carrying out in the testis zinc finger protein domain, and you can refer to this paper to understand in details how this work was carried out. Now, let us look into these figures. So, these are the zinc finger 1, zinc finger 2 and zinc finger 3, and as discussed earlier, they are wrapping the DNA molecule in a semicircle, as you can see over here. So, in this TJD DNA complex, the cognate DNA represented by cartoon with the phosphate backbone in yellow here, and the bases inside in violet, okay, and they are inside this molecule. TJD is also represented by a cartoon with the helix and surface mesh of zinc fingers 1, 2 and 3 shown in green, blue and red respectively. The beta sheet in each finger is omitted in this case for clarity. So, initially the C2H2 zinc fingers bind to the DNA in a non-specific manner, and then you can see that zinc fingers 1 and 2 gradually trying to interact with the bases in the center to find out its cognate sequence, and this type of condition is known as the semi-specific binding state. Now, this will keep on occurring until and unless the protein molecule find its cognate sequence. So, it always attempts to find out by reading or interacting with the bases in the core its cognate sequence, and if it fails to find it, so it will slide to a next location and try to read it, and this goes on and on until it will find its cognate sequence, and whenever that cognate sequence is being found, it changes to the specific binding already discussed to you. So, you can see that this is not a very simple reaction, it is quite dynamic, and it is a hit and trial effort or method after which a zinc finger finds its cognate DNA sequence. So, in the TJD DNA complex, the cognate DNA is represented by a cartoon as already told to you. Initially, the TJD interacts with the cognate DNA in a non-specific binding. The fingers 1, 2 switch from non-specific binding to semi-specific binding, with finger 1 almost fitting into the major groove completely, but finger 2 not fitting. Finally, the semi-specific binding is improved by the non-specific binding on finger 3, and the DNA binding is then transformed to the specific mode, with fingers 1 and 2 fitting into the major groove completely, but finger 3 binding non-specifically to the DNA. So, we now come to end of the discussion on the basics of zinc finger proteins as well as other proteins like FOC1, and together they are the important concepts for the zinc finger nuclease technology. So, now we know that the zinc fingers binds to DNA cognate sequences. By changing these amino acids, a high degree of selectivity can be achieved toward a given 3 base pair DNA sequence, and exploiting this recognition mechanism, protein modules containing multiple zinc finger motifs, each one recognizing a specific 3 base pair DNA sequence, have been engineered which bind to specific DNA sequences, and fusing this recognition module with a sequence independent endonuclease or DNA cutter was the first successful strategy to introduce breaks at specific sites of genomic DNA. And we have studied in detail about the double strand DNA breaks and how those are helpful in homologous recombination and other mutational events. So, in the next lecture we are going to discuss at length about utilizing these basic concepts in the development of zinc finger nucleases. Thank you.